Excellent. Our next presentation is by Dr. Angela Early from the Broad Institute. Broad.
Morning, everyone, and thank you to the presenters. Thank you for the organizers for putting together a great conference. I have been, and to the presenters, because I've been totally geeking out over the great diversity of research here. It's been a lot of fun. This is my first conference post-COVID and 2 babies, and so I'm just like back and feeling very excited to be talking science. So I'm a staff scientist in the Genomics Center for Infectious Diseases at the Broad, where I study malaria through an evolutionary genomics lens. And I have a particular interest in the human parasite Plasmodium falciparum in small populations. Small populations can be extremely frustrating to study evolutionarily because they can be governed by a lot more drift. Signals can be a lot noisier. So on one hand, you'd ask why, and from a pragmatic reason, it's just because we have a lot more populations that are now small. And if we actually want to prevent the persistence of Plasmodium falciparum and truly drive malaria to elimination, then we need to figure out a way to get it over that final hump and prevent these long-lasting persistence. Here you see how malaria gains have stagnated globally over the last 7 years. And the reason we can't just apply what we've learned in large populations to small populations is that the ecological and evolutionary dynamics are actually distinct in these 2 regions. It's fascinating. We have many hypotheses for why they diverge. I have a few up here now that I can't go into in full detail. But basically, these small populations maintain a really incredible amount of evolutionary adaptability and flexibility, and we see drug resistance preferentially arise de novo in small populations rather than large populations, which is not what you expect when, like me, you come from a Drosophila world where you expect evolution to be much more efficacious on a large scale. So what I'm going to focus on today is this bottom one here, and the fact that your effective recombination rate is different if you're in a small population with low transmission than a large population with higher transmission. And by effective recombination rate, what I mean is the recombination rate that we observe when looking at the population level. As many of you know, Plasmodium is a sexual diploid individual organism, but it has a haplodiplonic life cycle. Throughout most of its life cycle, including its entire life cycle within the host, it's haploid, and it goes through this extremely brief diploid phase following fertilization in the mosquito. So this means that if there are 2 distinct genotypes that the mosquito takes up in a blood meal, there will be an outcross in the mosquito. And as that mosquito then bites another individual, it will infect them with related but not identical parasites. In a high transmission region, there's a really high probability that this person will experience another infective mosquito bite. And so in the second generation, we can get another outcrossing event, and this cycle can continue with a nice mixing, and in other words, a high effective recombination rate at the population level. Now, in small populations, this rarely happens. Even if we start with this initial outcross event, the person who's infected by this recombinant progeny likely will not experience another infective mosquito bite. So we get rounds of inbreeding, sibcrosses, further inbreeding, until eventually there's really just a single genotype through these bottlenecks that makes it into a host. And therefore you get selfing in the mosquito, or really what amounts to clonal propagation, because this isn't even meiotic recombination through 2 different distinct chromosomes. This really is just meiotic recombination to identical chromosomes. And what we've seen in regions of really low transmission along the Pacific coast in South America is that these can persist for over a decade. We see multiple instances of 10 to 14 years, probably longer persistence. That's just what we've measured. And you can imagine this is changing the evolutionary dynamics a lot. And if you have a really high, a perfect suite, say, like the perfect storm of genomic variants within a lineage, this thing can just keep on persisting in the face of evolutionary change. This also changes how we have to measure evolution in these populations. A lot of our standard population genetic tools just simply don't have the power to give us much information in these populations. So one of the statistics we use a lot is to look at relatedness through a measure called identity by descent, which I'll walk through here quickly if this is a new idea. And this captures overall relatedness between individuals and also allows us to catch distinct recombination events, which we can actually look at as a measure of time or as a form of evolution, like a mutational event that we can track through these populations. So, and here we have a cross. Each individual can now be described, each progeny can now be described as a combination of genomic segments from the two parents. And if we line them up, we can see regions that were inherited from the same parent. And what we see is siblings typically have 50% IBD, 50% relatedness. In a second generation without crossing, this drops down to about 25% IBD. So what we see now, and when we go into actual populations with these measures, is this high relatedness. The case study I'm going to talk about today is French Guiana. French Guiana is up here on the northern coast of South America in this unique Guiana shield biome. And there's been a real marketed malaria elimination campaign in the 21st century, excuse me, which included here as well an institution in about 2007 of new drugs, insecticide-treated bed nets, further treatment coverage, all these things to really drive down numbers. And it was a tenfold reduction that you can see here. So we were interested to know what the genome is telling us about adaptation during this time period. This is work with our collaborator, Elise Musée at the Institut Pasteur in French Guiana. She leads a great team who does not only sample collection, but parasite adaptation, phenotyping, targeted genotyping of these samples that we were able to leverage. So what I'm going to focus on today is what's happening on either side of this drug regime change that happened in 2007 with these longitudinally sampled parasites. And not surprisingly, just what we would expect from theory, but it always surprises me a little bit when we actually see in the data what we expect to see, is that relatedness increased through time. We had a large bottleneck, and this really does show up in the genome. And in fact, the lowest relatedness we see in 2013, 2015 is above the median relatedness that we saw before this drug regime change. So this shows the successful population contraction. And if we look more discretely within the genome, we see that there's a lot of variation. So it's not just that there's a straight line of variation across the genome that moves up as this population shrink. It's really unevenly distributed. Some of this is surely due to drift. Without a doubt, I think I actually have a duplicated slide here. Okay, some of this is surely due to drift, and we need to understand the demographics of this population more to know how much of this could just be background stochasticity. But we also see evidence of both positive and purifying selection. We see purifying selection because we see greater changes in non-coding and intergenic alleles that probably have just drifted more, and so greater conservation of coding alleles. And we also know there has been selection across this time period due to phenotyping led by our collaborator, Lise. So this region of South America, like globally, experienced really strong chloroquine pressure throughout the 20th century, to the extent that this resistance allele in the CRT gene, K76T, had fixed by the 1990s. So then, not surprisingly, French Guiana moved off of chloroquine into a different drug regime, quinine doxycycline. And switching this drug then exposed the fitness cost. There's a well-documented fitness cost to this K76T mutation. Now, when this same experiment has been done in Africa, these larger high transmission populations in Africa actually saw a reversion then to the wild type K allele at the CRT locus and a return to chloroquine susceptibility, again, because of this fitness cost. But in South America, this allele had fixed. There was nothing to revert to. So these parasites were just kind of left at a lower fitness. And on another talk, we could certainly talk of and debate, okay, what is relative fitness in these different high and low transmission regions? But nevertheless, there is a fitness cost. But nevertheless, chloroquine susceptibility started returning phenotypically within this population. So first, I want to stress that Plasmodium persists, okay, the theme of this conference, this session, but also that there was some sort of change there without this reversion, this allele. And what work that Lise did with, oh gosh, I need to go faster. Our previous collaborator is that there was this genotype change at C350R elsewhere in the CRT gene. So what we further did then was, knowing that these populations can still adapt, dug more into the details of the genome using a haplotype test. This is an H1/2 statistic. Can talk more about genomic tests with people if they want to do later, but what I just want you to know here is that higher values means higher evidence for selection, lower values, lower evidence for selection. Here we have the 14 chromosomes of the Plasmodium genome and windows going across. So this is before drugs were changed. We see nice peaks here. This is just for benchmarking the 95th percentile up here. And I'm marking here genes that are known globally to be involved in drug resistance that could be implicated here. And we see some nice results where we expect based on what we know in the South American population. We also see genes that are implicated elsewhere, but had never been implicated in South America before. So this is an interesting result to see. It expands our knowledge of what's going on in this population and novel peaks that obviously are going to be followed up on to expand our idea of how drug resistance has emerged. Now, the important thing about this temporal sampling is that what we see in the later time periods after a new drug has been added, and this is the type of sampling we would typically do, is just go out, sample things and look at them. We see the persistence of a lot of the peaks that were actually from the previous gene, not the newest change in 2007 to a previous drug, not the new drug that was introduced in 2007. And this could is important then to have this temporal idea to know what is new, what is old. In this case, these aren't going away. These are just lingering signals in the genome. But because of the temporal sampling, we can show which one now are new peaks. And we see two really distinct new peaks that did arise with the change of drug regime in 2007. Now, what is missing here, so there's a lot to focus on here, but I'm just going to focus on this one thing that's missing is the CRT signal that I talked to you about, where we had basically a sweep upon a sweep from the 70s and then another sweep in the early 2015s. This is a very soft sweep that we're finding hard to detect with our typical genomic measures. So we know there was this huge change in frequency, a 60% change from 2000 to 2012. I mean, this looks like really strong evolution that's happening. But this C350R mutation is on 3 different haplotypic backgrounds, not just one as you would expect with a hard selective sweep. So what I'm showing here instead, though, is we still see patterns in the genome, but it's a different type of pattern. And again, we can only see this with this temporal sampling method. But instead of seeing selection for a novel haplotype, we're actually seeing selection against the old haplotype. So this is a haplotype that was nearly at 50% at the CRT locus before drugs were changed, and it's completely dropped out of the population now that we're in a new drug regime. So in conclusion, I'd just like to highlight that Plasmodium populations successfully adapt, even when they're small, even when they're close to elimination. These are persistent parasites. And the other aspect of persistence we see is in these drug mutation genes themselves. They can fix in these populations, stick around, and maybe revert, but also stay there probably because drug pressure is different in these regions. But there's a good side to this as well, and ways we can change our methods for discovering these signals. First of all, temporal sampling is key. I think samples should just be taken, and even if not money for sequencing, just keep them so that as things become cheaper, we can continue to do this temporal analysis. And secondly, these very soft sweeps that kind of become stacked on genes that we know are important for multiple drugs are going to be important to really dig into and understand with new methods. So with that, I would like to thank Dan Eastie's group, which is based at the Harvard School of Public Health, as well as the Broad Institute, our collaborators throughout the Guiana Shield area of South America, and of course, the patients who helped us learn these things. Thank you.
Question up in the back.
Thank you. Hi, that was a great talk, really interesting. I actually have 2 questions, if that's okay. The first one is that you talked a lot about recombination, but even in a low transmission setting, an individual infection is going to have a really high number of parasites in it with lots of de novo mutations. So what do you see as the role of de novo mutations in the evolution in this kind of setting? That's question 1. And question 2 is, given all the social and economic and political changes going on in the Guiana Shield region right now with Venezuela, are you expecting to see a rapid transformation in the malaria landscape and resurgence of malaria?
Oh, these are 2 of my favorite questions. Thank you so much. De novo mutations is something I'm very interested in digging into more in these populations. There's large bottlenecks coming in and out of the host and the mosquito, so that does constrain this some. But certainly when you see the same mutation on the background of these different haplotypes, this is one of my leading theories for what is happening and an underappreciated source of evolution in these populations. So we're looking, if anyone has ideas of how to better dig into this, I would love to chat with people. And then as for what's happening specifically in Venezuela, yes, and this is being monitored throughout the South American region. Again, this IBD approach is a perfect way to go after it because we can actually see what chunks are held in common between Venezuelan parasites, Pacific Coast parasites, and Guiana Shield parasites, which historically over the last 200 years have been rather distinct. We can see genetic differences between them, so we actually can track introgression, and there's a lot of work going on in that area. Yep.
Thank you very much for an insightful presentation. We spoke about the importance of temporal tracking, right? And so what part does spatial tracking of resistance also play in this? That's one. And then for the second one, do you have any idea why transmission always comes up from, resistance always comes up from the low transmission settings like Southeast Asia, South America, as compared to Africa, where transmission is high, but you don't usually have resistance coming up from Africa? Yeah.
Yes. So for the spatial sampling is also very important. And again, if we as a community want to understand more whether infections, for instance, are imported into a region that's nearing elimination versus being an endemic transmission, the only way to do that is with good spatial sampling. One thing, again, we can do with this IBD approach is not only say, okay, this whole parasite looks like a background from another country, but we can say this region around this drug resistance allele looks like a haplotype from another country. And in the South American region, specifically, the drug resistance mutations or backgrounds, haplotypes in Venezuela are in fact distinct from the ones we see in Colombia and Guyana, making that possible to track. Again, we need good distribution of sampling, so that's important. As for why resistance arises first in these low transmission regions, there's a lot of good hypotheses out there that certainly hold up with some modeling and some data. I would say no doubt it's a combination of a lot of factors. Some of the big ones would be different immune selection that's happening. The immune pressure is going to be much less in low transmission areas. They're going to confront drugs more at a higher rate because there's less asymptomatic infections in those areas. There's also the idea that you can have better epistatic effects across the genome because of different recombination dynamics, and the list kind of goes on from there. But jury's still out on that, I think.
Excellent. Thank you very much.